Hi, my name is Margo McBrearty. Welcome to my presentation for the Global Education Symposium. My name is Margo McBrearty. I am an undergraduate biology major in the College of Science here at RIT. I have focused and participated in viral research during my undergrad. My international internship during the summer of 2021 was a virtual Mitacs Fulbright Globalink internship with a professor at the University of Montreal in Quebec, Canada. I was also lucky enough to be able to continue working with my supervisor, Dr. Bargest, through this fall semester as a virtual visiting student researcher. Here is a quick table of contents for my presentation. We're going to start with the internship overview, then I'll touch on my literature review, the review paper I collaborated on, the laboratory website I created, and then finally my main project, which was near-deep novel microRNA analysis. I interned in the Bargeste Immunology Laboratory located in St. Hyacinthe, Quebec. However, due to the pandemic, it had to occur virtually, which changed what an international internship experience was like. Regardless of the virtual nature, I was still able to experience working with people from around the world who had all taken different paths to end up working in this lab. It was an amazing opportunity to gain experience working amongst multiple time zones on a collaborative project, and the Bargest lab works on antiviral responses in chickens while infected with viral pathogens. Their aim is to elucidate what mechanisms the chicken immune system employs to fend off viral infections and what regulatory mechanisms are in place in the respiratory system specifically. My supervisor was Dr. Nada Barchest, who is a PhD and a doctor of veterinary medicine and currently working at the University of Montreal in Quebec, Canada. I performed a literature review that entailed searching and reading through all the current scientific publications that were focused on what microRNAs play a role in or are affected by infectious bronchitis virus infections. Then I compared these findings to the research my supervisor already accomplished on microRNAs that were found to be affected or impact avian influenza virus infections, to identify which microRNAs were found to be in common. The figure on this slide shows 3 examples of microRNAs that were found in common between avian influenza virus on the left and infectious bronchitis virus to the right. GG microRNA 1563 was seen to be upregulated in both pathogens. Then microRNA 146A5P was found to play a role downregulating the innate immune response during both infections. And finally, microRNA 6516 5-phosphate for avian influenza and 3-phosphate for infectious bronchitis was hypothesized to be connected to IBV's virulence, and in AIV, this microRNA was also seen to be upregulated. I worked on a review paper with the topic of microRNAs in chicken infectious diseases, moving forward towards innovative approaches. My categories were infectious bursal disease virus, infectious bronchitis virus, coccidiosis, and Cryptosporidium Bailey infection. I was able to create a laboratory website for my supervisor. The website included things such as the current news, current research, recent publications, and the laboratory members. The aim is to increase the reach that her research, but in a broad sense, all scientific research has. It aims to increase the public awareness in new scientific research and also increase the general public's confidence and trust in science. The process I took to create the website was first purchasing a domain name and then purchasing a hosting service and then designing and filling the website with my supervisor's wanted content. The general topic of my internship was microRNAs. I was focused on identifying what novel microRNAs might be connected to the immune response in chickens, what regulatory roles they have, and how their interaction with mRNA affect the antiviral response in chickens affected with avian influenza virus. So what are microRNAs? MicroRNAs are small RNAs, only about 18 to 22 nucleotides long. They're non-coding and they're conserved. They're gene silencing through either translational repression or targeted degradation. And this is seen in the figure to the right, where you see the microRNA binding to the 3' untranslated region of mRNA transcripts, which either leads to translational repression or mRNA degradation. And this affects the expression of a specific gene or gene pathway. So, microRNAs are thus seen as regulators of the antiviral immune responses. My project was a continuation of the research seen on this slide. This slide shows the prior experimental work that resulted in the characterization profile of known microRNAs for avian influenza virus, which is denoted with the 1, seen underneath the small RNA sequencing. My focus was to characterize the novel or unknown microRNAs that's denoted with the 2, also under the small RNA sequencing. This slide depicts the results from the methods that were visualized on the previous slide that worked in characterizing known differentially expressed microRNAs induced by avian influenza infection. By seeing the known microRNAs being characterized, it shows the gap in our knowledge and the microRNA databases of novel microRNAs in chickens in particular. The LSP and poly-C are ligands that were used as a form of positive control, as they had been reported to induce immune response in the tracheal epithelial cells of chickens. The rationale for this project was as followed. Host pathogen interactions and the induction of antiviral responses is analyzed, and microRNA regulation of antiviral responses during viral infection. We were looking into what specific targets and roles that microRNAs play, and specifically for my project, looking for unknown microRNAs playing a role in regulating the antiviral responses. These studies are required to determine the mechanisms by which cellular microRNAs influence and regulate antiviral responses, and this allows us to develop new approaches or agents to fight viral respiratory diseases in the poultry industry. Here are my hypotheses and objectives for my project. We hypothesized that there are uncharacterized microRNAs regulating the antiviral response against avian influenza virus infection in chickens, and the objective was to identify the profile of unknown microRNAs upon avian influenza virus infection of chicken tracheal cells. Here is a visualization of the methods used to identify the novel microRNAs. First, a small RNA was sequenced using Illumina HiSeq 2500, and then adapter, trimming, and quality control was performed. Then, these alignment of reads to the chicken reference genome and to the database of known microRNAs was done, this leading to identification of novel microRNAs as well as identification and quantification of the differentially expressed known microRNAs. This was using the miRDeep2 software. So what is miRDeep2? miRDeep2 is a prediction and quantification pipeline written in a Perl language which can identify both known and novel microRNAs from deep sequencing data. But why is miRDeep2 used or trusted? Well, it was proven that it can identify with high accuracy and sensitivity known and novel microRNAs in all species except for the fly. And then determining criteria for genuine microRNAs from miRDeep2 analysis must fit any or all of the given criteria, greater than 4 on the miRDeep score, no match with RFAM, a significant RNA fold, and greater than 10 mature reads. Here is an example of the miRDeep2 program results when ran with the tutorial analysis. This is an example of the output data for cell microRNA 40. The red part of the sequence is the identified region that varies from the normal sequences. So the future use and direction that this project can go in is in one, experiments. The next experiments done with this data will be in identifying the novel microRNA's functions using functional analysis in vitro, as well as comparing the novel microRNA's function to the known microRNA's function, also in vitro. And then this data can be applied by employing newly found microRNAs as adjuvants for vaccines. Adjuvants are a way to increase the immune response and thus the efficacy of vaccines. The end goal with this research is to lead to vaccines with increased efficacy in order to protect the poultry industry workers as well as the poultry itself from infectious respiratory viruses. And lastly, I want to say a great big thank you to first Dr. Nada Barchest for giving me the opportunity to work in her lab this past summer and allowing me to continue working in her lab this fall semester. I'm grateful for the opportunity to see the review paper through to its completion and have a chance to give a presentation to her lab department. I learned valuable communication and bioinformatics skills during this internship, and I also learned a lot about viral research and virology through the work I did. Dr. Barchest also gave me the opportunity to take a Coursera course on the basics of extracellular vesicles that was taught by leading professors around the world. This helped to supplement some of the knowledge I would have learned had I been able to be in the lab physically. And secondly, I owe a big thank you to the RIT Global Department for providing me the information I needed to apply and take part in this internship. I specifically want to thank Jenny Sullivan for her continued help and support during the application process. Thank you, and I hope you enjoyed learning about my international internship during the summer of 2021.